Okay, we have 10 o'clock, so we can slowly start. Probably a few more people will join us soon. So welcome to the very first installment of the Polish NMR seminars organized by the section of Magnetic Resonance Polish Chemical Society. So first I would like to show you few things, one slide about the idea of this seminar. So the speakers should represent the Polish NMR community, and we define this community as someone fulfilling at least one of these conditions. So be a Polish citizen, conduct research in Poland, be an alumnus of Polish Research Institute or university, or have very close cooperation with Polish researcher, and you want to share these results with the community. Yeah, we are open to the speakers at every stage of the career, so we are very welcome to postdocs and students, to not only PIs. I'm actively looking for speakers, so please let me know if you have some research to share with us. And I would like to have a
signal,
special...
but a signal of your macromolecule. So
I think I need to mute Jens Nowak. Okay. Yeah, and there is a special offer for students. If you want to make a short presentation, like 10 to 15 minutes, we can squeeze you between the main speaker and we can have like 2 talks at once. So many people have problems filling like full hours. So if you're a student and want to have a short presentation, share your research, please contact me. So with that in mind, I'm now giving the stage to Krzysztof, who will be our first presenter. So Professor Krzysztof Kazimierczuk from Center of New Technologies, University of Warsaw. And Krzysztof, the stage is yours. You can share the screen again.
Thank you. I'll try. Okay, can you see it well?
Yes, we see it now.
Okay. Okay. So I hope also you can hear me well.
That too.
Okay, good. Okay, so it's my pleasure to give this first seminar in this series. I am Krzysztof Kazimierczuk from the University of Warsaw, from the scientific unit called Center of New Technologies, focused solely on the research and purely grant-based. My research interest covers mostly the signal processing and signal acquisition methods for NMR. And well, the topic of this seminar, the title of the seminar is very long, as you can see, because my intention was to cover the majority of our activities over the last, let's say, 2 or 3 years. So I decided I will refer very briefly each of the papers that we published, and the details you can find in the references provided. So we mostly work with small molecules and their mixtures. The small molecules, as you know, are often simple in structure, but it doesn't mean that their spectra are necessarily simple. Here, for example, you have a spectrum of sucrose of natural origin, which is slightly hydrolyzed. So besides sucrose, you have glucose and fructose. You have significant peak overlap. You have, well, quite a big range of signal intensities, and all of this leads to quantification and identification problems, even in simple mixtures like this. And moreover, after several hours in the spectrometer, the spectrum looks like this, because sucrose hydrolyzed even further, some of the alpha glucose converted to beta glucose, shims got distorted, as you can see. So we have line broadening, which is not simple because it's not non-Lorentzian and non-Gaussian broadening. So the problems with picking, for example, they arise in time. Okay. As you know, there are many solutions to these problems. One of them is to resolve components physically. For example, with chromatography that can be coupled to NMR spectrometer or performed independently. You may resolve the components of your sample in pseudo-dimensions, like diffusion pseudo-dimension in dosy spectroscopy or relaxation pseudo-dimension in rosy spectroscopy, and so on. You can try to improve frequency resolution by introducing extra dimensions to your spectra, 2D, 3D, and so on. Here, many techniques of alternative sampling, like fast sampling or non-uniform sampling, can be introduced to accelerate the measurement or further improve the peak resolution. Or you can try to improve the analysis by introducing various deconvolution methods that will deconvolve these peaks, try to separate them, separate components of your mixture into subspectra of individual components. The methods that we developed recently fall into these two categories. So we worked on introducing new pseudo-dimensions into NMR spectra, and we worked on the new methods of analysis. So in my talk, I am going to cover the following topics. So first, I'm going to talk about pseudo-dimensions like temperature, pH, and solvent. And then I'm going to switch to advanced methods of mathematical analysis of the spectra based on Wasserstein metric and numerical shimming or correction of distorted spectra using machine learning. So all these factors, temperature, pH, and solvent, as you know, they may affect chemical shifts. And the fact that changing temperature or pH or solvent composition may affect chemical shift can be exploited to resolve peaks or resolve peak overlaps and to support assignment because it is quite specific, as I'll show in the next slides. So first topic, temperature pseudo-dimension. Application to mixture analysis, specifically to analysis of samples of metabolites, but not only. So if you acquire a spectrum of a mixture of metabolites, like blood plasma, for example, or urine, and so on, and you vary the temperature, you will see that peaks shift in quite a specific way. So not only chemical shifts are specific to given compounds, but also the way they change with temperature. These changes there are quite slight, but as we showed in our paper in RC Advances referenced here, they are reproducible between mixtures and they are quite specific. So although you cannot use them alone to assign peaks, which wouldn't make sense, you can use them to support your assignment. Especially cases like differentiating between creatine and creatinine is easier using temperature coefficients. This data, such series of spectra with varying temperature, can be processed using radon transform. The radon transform is a method introduced to NMR by Kupche and Freeman over 10 years ago. And the idea is quite simple. When you have a series of spectra where peaks shift linearly, you can treat this series of spectra as one two-dimensional object and calculate the projections at different angles, literally geometrical projections of this data. And when the angle of the projection matches the angle at which given peak moves, the summation or projection leads to, well, amplification of the peak, just a direct summation. When it doesn't, then you get a broad blob. So you calculate a set of projections and they form a new two-dimensional object where you have peaks, and the peak positions, they correspond to chemical shift or frequency in one dimension and the rate of change of chemical shift in a series in the other dimension. Of course, these peaks, they are not Lorentzian peaks. They have this strange butterfly shape, meaning that picking them with standard programs is not necessarily simple, but we developed own peak picker that I will show on the next slides. The extra advantage of radon transform, besides giving you direct access to these rates of change of peak position, is the gain in signal to noise ratio. Here is the extreme case. So even if you don't see peaks in the individual spectra, only noise, the projection or coherent summation of peaks allows you to see them. So you see, for example, blue projection is the projection that matches the angle at which right peak moves, and you see it in the projection even if you don't see it in the individual spectra. So radon transform gives you the signal to noise ratio boost. So as I said, we developed the radon peak picker, the program that can peak, that can perform radon transform and peak the series of spectra. Here is one of the examples from the metabolite sample. So you have creatine and creatinine, two singlets that are hardly visible in the individual spectra. Almost nothing can be seen there, but coherent addition of this 20 spectra gives you indeed two singlets, and they differ in their positions in the radon dimension. So they differ in their so-called temperature coefficients, or in other words, the rates of change of chemical shift with temperature. So you can, thanks to this trick, you have extra factor characterizing your peaks, because as I showed before, these temperature coefficients, they are reproducible between mixtures. In our paper, we showed the reproducibility between blood, between artificial mixture of metabolites, and between extracts, biological extracts from biological tissues. Okay, here you have another example, choline singlet next to the multiplet, singlet, all triplet components, they have the same temperature coefficient, but singlet next to it has different temperature coefficients, and it can be recognized. And also the signal to noise ratio boost is there. The same with alanine here. So we have alanine doublets. If you are not sure whether it's a doublet or two singlets, you can perform radon transform, and you see that they have exactly the same temperature coefficients, meaning that it's rather doublet, not two singlets. Because, well, of course, unless J coupling changes with temperature, which is not so common. Okay, so second example of application of this temperature pseudo dimension is a resonance assignment transfer between similar compounds. So we had the following NMR problem. 7 derivatives of this compound have been synthesized by our colleagues, and they wanted to assign the spectra. The question was, can we somehow use the temperature coefficients to transfer the resonance assignment from product 1 to other 6 products? So we assigned product 1 using a standard set of two-dimensional spectra, like HSQC, HMBC, NOSI, and so on. And then what we did, we measured proton NMR spectra with varying temperature for other products. And well, what we observed was that temperature coefficients were more specific than chemical shifts. Of course, the differences in temperature coefficients, they are small. These temperature coefficients here, they are in ppb per Kelvin, so they are very small but measurable. And as you can see, for example, for this doublet of doublets, chemical shift varies between different products, different derivatives of this compound, but the temperature coefficients stays much more constant. These plots show temperature coefficients and chemical shifts for protons of these compounds for different products. And as you can see, the 4 aliphatic protons from the middle of this molecule, in all products, they form similar pattern, right? So, of course, you cannot use variable temperature proton spectra alone to support, to assign your molecule. But if you assign these products using limited set of 2D spectra that still leaves some ambiguities, you can find them or even resolve them using variable temperature spectra. So also in other regions, for aromatic signals, there are certain patterns, for example, proton Y, so the one that is here, close to the substituent. So its temperature coefficient is very well preserved between samples, contrary to chemical shifts that is heavily dependent on the substituent, on the derivative. So we think that variable temperature NMR can be used to support resonance assignment, although it's not easy to model these changes. But in some cases, as this one, when you have a series of similar compounds, you can detect probable misassignments or support the assignment using variable temperature spectra and comparing temperature coefficients. Okay, finally, we use the same idea in protein NMR to support the assignment of intrinsically disordered proteins. These are not small molecules, so this is beyond the topic of this seminar, but I'm just going to mention this quickly, our paper. So in proteins, the chemical shifts are primarily dependent on the residue types. I mean, in intrinsically disordered proteins that lack stable structure. So the spin systems can be quite effectively assigned to residue types based on chemical shifts of amide protons, nitrogens, carbonyl carbons, and so on. And this classification can be effectively performed using linear discriminant analysis, as we showed in our recent paper. And moreover, as we showed recently, adding temperature coefficients to chemical shifts, so extending this set of characteristic factors, improves the analysis. So in other words, these patterns that I showed here, they can be analyzed statistically using advanced methods like linear discriminant analysis. But we did it for proteins where we have much larger databases which can be used to train the method. For small molecules, it's impossible. Only quantitative visual analysis is possible. So, okay. So another idea for new spectral pseudo-dimension is use pH as a pseudo-dimension. To allow this, we had to use the device developed by Darek and Agustin from my group several years ago. So this device is so-called SWAPE, sweeping apparatus for polarization enhancement. And this is a simple step motor that you place on the top of benchtop spectrometer, and that allows you to shift the sample vertically. And this shift can be synchronized with the pulse sequence. So this allows you to shorten D1 because you can basically excite volumes of the sample one after the other and not wait for them to relax, just shifting the excited part of the sample outside the coil region and switch to another volume. So this is perfect for quantitative NMR, especially when you have to deal with long T1s. And also it opens way to gradient pseudo-dimensions. Now, what do I mean by gradient pseudo-dimensions? So the idea is inspired by the paper of Matthew Wallace, published several years ago. What they did, they created a gradient of pH along the sample tube, and then they used chemical shift imaging or slice selective experiments to separate small slices of the sample and measure pH-dependent chemical shifts. Now, what we did, we used the same or similar sample preparation method, but ran experiments in different way. So we physically moved the sample using SWAPE device and recorded spectra of consecutive sample volumes that correspond to different pH. So the sample is created in a very simple way. You basically put some acid at the bottom of your tube, and then you gently add the sample without mixing it. After 15 minutes, you have nice and stable pH gradient. What is the use of it? So, well, it is spectral pseudo-dimension. So it has the advantages, some advantages of 2D spectra. So using this pH gradient allows you to boost resolution. For example, here you are able to resolve peak of taurine in the natural mixture, which is an energy drink. And you are also able to characterize this peak by additional factors. So not only chemical shift, but also the way the peak position changes with pH. Here is the example of contaminated powder milk, contaminated with melamine, and only at low pH you see this melamine peak appearing. Now, our recent research is focused on solvent pseudo-dimension. So similar idea, but with solvent gradient along the sample tube. Here is the example of our study of naproxen tablets, so commercially available medicine. And you have active substance, so naproxen, and some extra substances like lactose, for example. Now, placing this mixture in the solvent gradient, like a mixture of DMSO and D2O, and running a series of spectra with the use of SWAPE allows you to resolve the components. So in DMSO, you see mostly naproxen, while the more water you have, the less naproxen resonances you see, and you see the lactose peaks appearing, which are covered by naproxen peaks in the DMSO spectrum. And well, the most advanced application of SWAPE, of the device shifting the sample vertically, is our recent split inversion recovery experiment. So the idea is that you can, that in inversion recovery experiment, which involves 180-degree pulse followed by recovery delay and 90-degree pulse. In this experiment, you can separate the inversion pulse and read out 90-degree pulse and FID acquisition by the sample shift. So you can apply 180-degree pulse to one sample volume, then shift your sample, apply 180 to another volume, shift it, apply to another volume, and so on. Then go back to the first volume and apply 90-degree pulse and FID acquisition. And this allows you to accelerate the experiment because you don't have very long passive delays. Of course, it cannot be done for very small tau values or very small recovery delays. So first 4 points are acquired in almost conventional way. So first 4 taus are sampled using conventional inversion recovery. We just shift the sample between consecutive acquisitions of inversion recovery to shorten D1. And then we switch to the actual split inversion recovery. Okay. So for example, here you can see that we apply 180-degree pulse to volume number 2, then to other volumes, and then we are back at volume number 2 and apply 90-degree pulse and FID acquisition. So here are the results for the sample of formate that is quite commonly used standard for quantitative NMR, and the results of SWAPE, they match quite well with the result from conventional inversion recovery, but the acquisition time is 5 times shorter. So that's all about pseudo-dimensions. Now I'll switch to improved analysis. So first, the application of Wasserstein metric. Together with a group of mathematicians from the University of Warsaw, we developed the approach called Magnetstein, which is a very useful tool for quantitative NMR of mixtures, reaction monitoring, and so on. So the idea is as follows. In conventional approach to quantitative NMR, you need to pick your spectra, which leads to problems when you have very low resolution, like here, for example. So here we have a benchtop spectrum, 43 megahertz, proton NMR of certain dietary supplement involving 3 amino acids. So this supplement is basically a mixture of valine, leucine, and isoleucine. Okay. Now the low resolution leads to problems with picking, which heavily biases the quantitativeness. Now the idea is to skip the picking and treat this spectra as literally as piles of sand. So this is how Wasserstein metric works. Wasserstein metric is basically the minimum total cost of reshaping one pile of sand into another pile of sand. And by total cost, I literally mean the work needed or the fuel consumed by the bulldozer that reshapes one pile of sand into another. So how to apply it to analysis of mixtures? Well, you need to build a library of spectra, of spectral components that can be possibly present in your mixture. Then we build linear combinations of the spectra from this library, and then we calculate then we look for the minimum Wasserstein distance between this combination and the mixture. Okay. Now, the advantage of this approach is that first, it doesn't require picking, and second, it is insensitive to distortions of peak shapes. So there is no assumption about peak shapes, like whether they are Lorentzian, Gaussian, or anything. The spectra can be even heavily deshimmed, as I will show on the next slides. And second, the Wasserstein metric is sensitive to the peak shifts. So the Wasserstein distance allows for small differences between spectra in the library and the spectrum of a mixture, and you don't have to assume here any maximum shift, any radius, and so on. So here is the result for that supplement. With colors, you see the contributions from different components, and the Magnetstein finds right proportions in this supplement. It is quite robust to peak shifts and line strip distortions. We show many examples of the applications also to the high field spectra. In our paper in analytical chemistry, we show how Magnetstein allows you to deal with problems with shimming, differences in temperature between library and mixture, and so on. Okay, here is our recent work involving Wasserstein metric or Magnetstein. So we have reaction monitoring problem. We have a series of spectra of reacting sample, the hydrosilylation, shown here. There could be no deuterated solvent used, so no log. There was no time for shimming because the reaction started rapidly after injecting the initiator. And also temperature has been stabilizing during the measurement. And that's because the sample had to be kept in liquid nitrogen just before starting the reaction. So in the consecutive spectra, we see heavy line shape distortions, also peak positions change. And this, of course, biases the peak picking. The question is whether monitoring is impossible in such cases. Well, it can be done much better using the Magnetstein approach. Here are the results for this reaction. For Magnetstein processing compared to simple integration in MNOVA program, where the integration was biased because peaks of product and substrates, they overlap, and actually it was impossible to find even a single well-resolved peak of the product. And this overlap, of course, naturally biases the kinetic curves. So you see the product curve is too close to the substrate curve. While Magnetstein, that doesn't require peaks to be resolved, doesn't require peak to be Lorentzian, deals much better with such cases. Okay, now the final topic that we work on is the shimnet. So in cases where you have spectra distorted by various magnetic field inhomogeneities, and you cannot repeat the measurement, you may try to correct spectra using some dedicated processing. The most known method of this kind is reference deconvolution, but reference deconvolution requires you to guess the parameters of the well-shimmed peak, and also you have to point at well-separated peak that can be used for reference. We tried different approach. So what we tried was to create a neural network that could learn the magnetic field inhomogeneity specific for the given spectrometer. So we take the calibration sample, for example, well-known line shape sample, the chloroform in deuterated acetone, and we measure a series of spectra with intentional shim deformations, okay? Series meaning thousands of spectra. So we do it in a loop that takes hours. And then having these experimental deformations specific for a given spectrometer, we generate synthetic spectra and we teach the neural network how distorted spectra on this particular spectrometer may look like. And then this trained neural network can be used to correct samples from routine measurements. Of course, and without any extra assumptions, except that they've been measured on this particular spectrometer and that they are not distorted more than the spectra during training. And now for slight deformations that are met in routine measurements, this tool works quite well, as I'll show you soon. So the neural network is, well, it has auto encoder structure. So it is built of encoder followed by decoder. There is also attention layer, and this attention layer selects peaks in your spectrum that reflect the distortion present in affecting all peaks. So basically, highest attention is paid by the network, is paid to narrow singlets. As you can see here, this is one of the spectra used during training, and the highest attention is paid to peaks that are best resolved and have the most pronounced dimming effect. So training looks like this. We generate spectra using quite simple but effective ambit NMR software. So we feed it with 10,000 of SMILES codes from an NMR database that are, well, typical small molecules. And then we take random multiplets from this output of the software, and having these multiplets, we generate synthetic spectra, placing these multiplets at random position. We convolve them with experimental response functions measured in the mentioned calibration loop. And this is how we get distorted spectra for training. Specifically, we used 40 million distorted spectra using over 10,000 calibration measurements. So after training the network, which has to be done only once for a given spectrometer, we can use this network to correct spectra, and the use is very easy. It's single click, no parameters. So the idea of the method is that the staff can calibrate the network or train the network for a given spectrometer once using well, the day-long measurement on the line shape sample. Then the network can be trained, and people are running routine measurements on this machine. If they didn't shim their spectra perfectly, or they forgot to adjust shims between samples, or some other distortions, slight distortions came, they can correct the spectra with a single click, just applying the trained network to their spectra. Here are two practical examples. One is that we have a sample shifted by 0.5 millimeter in the spinner and not reshimmed afterwards. So you see the shims are distorted. Here in the middle spectrum, the line shapes are affected, they are broadened. They are not heavily broadened, but they are broadened. And then you can apply the trained network, the ShimNet, and it will correct your spectra since it learned how typical distortions look like. Another example is the sample volume decreased by 20 microliters, and reshimming is not applied. It's just a volume decrease and a measurement performed again. Now ShimNet corrects this spectra. It even gives you, in some cases, gives you a result even better than for optimum shims. It finds the substructure of these triplets here. But generally, the line shapes get corrected. Some residual artifacts are present, of course, but generally the spectrum looks far better. And the method has no adjustable parameters, so it's easy to use even for non-experts. Okay, so I would like to finish by thanking all people involved in all these projects. I hope I listed everyone. well, everyone in this slide was at some moment the employee or a student of the University of Warsaw. So I can say that, well, all of them worked somewhere here around in my campus. So I didn't provide any affiliations, but currently, of course, for example, Mateusz or Darek work in Polish Academy of Sciences. Agustin Romero is back in Argentina. And well, people move, but generally, I think to the University of Warsaw. The research has been funded by National Science Center with Opus 18 and Opus 23 grants. And with this, I would like to thank you for your kind attention.
Thank you, Krzysztof. So we can open the discussion. If you have some questions, just turn on your microphone and, if possible, camera and start asking. So maybe in meantime, I'll ask one question. For your VT NMR measurements, do you need the temperature to stabilize or it's out of equilibrium during the measurements?
Well, actually, there are different approaches. So for the cases that I showed, there have been stabilization period. But another area of my research that I didn't mention here is so-called non-stationary NMR, where we measure signals that are out of equilibrium. So the frequencies change during the measurement, which is mostly applicable to 2D spectra, not 1D spectra, where changes affect the indirect dimension. But for the cases I showed here, all the spectra, they require stabilization periods, of course.
Any questions from the audience? Oh, Professor Potrzebowski.
Krzysztof, if I may ask the question. So what you see a feature for this ShimNet approach? So it will be commercially adopted by Georg-Brucker companies. So it will be only used by group who cooperated with you.
Well, we didn't talk to Brucker or Geo so far. That's what I can say. I don't know whether they'll be interested or not. I would say this is not only about introducing the program into their software. It is also about calibrating the network. So you need to introduce an extra calibration procedure, probably while installing the spectrometer, or well. So I'm not so optimistic about introducing it into commercial software, because they are often quite reluctant to this kind of modifications of their standard procedures. But who knows? There is at least one similar approach developed by Chinese group. I don't remember the affiliation now, but it also involves neural network to correct the spectra distorted by magnetic field inhomogeneities, but it requires much more user involvement. So it requires picking of this distorted spectra, similarly to reference deconvolution method, and it requires separate training of the network for each individual spectrum. But it can deal with much heavier distortions than ours. So we can deal with quite slight distortions that are typical for the cases where you have quite similar samples, but you have small distortions during measurements. So you can imagine the case where you have a sample changer and you just run 20 samples, but you didn't reshim between samples. So they had similar volumes, that's fine, right? So the solvents were quite standard and samples are homogeneous, but you just didn't care about shimming. Then ShimNet can deal with such slight changes. But well, knowing how it works, we will probably have to popularize this method on our own, and then maybe some big fish will get interested. We will see. For now, it's in the paper is almost ready. I hope we will submit it within weeks.
Anyone else with questions?
Me, hi, Paulina. I have a question to this topic. And what about the different samples? For example, you have calibration on particular sample, or this calibration could be, moved to another samples.
You mean shimming? You have shimming. Or you mean you mean the no. Okay. So back to this slide. So calibration is performed using line shape sample. So for example, chloroform in deuterated acetone, the standard light shape sample. So you don't have to calibrate the network separately for each sample. So you perform calibration only once for a given spectrometer, and then you use the trained network to correct spectra of different samples. The only requirement is that the inhomogeneity profile, so to say, of the spectrometers should be preserved. So you should train the model for a given spectrometer. But then the model can be used to correct spectra of many samples. So the idea is, I will repeat this maybe, the idea is as follows. The technical staff trains the network for a given spectrometer. So performs the calibration measurement that takes tens of hours, collects thousands of spectra. Then they perform the network training. And then they provide this trained network to the users, to the routine users, inexperienced spectroscopists that collect spectra on a given machine and sometimes make some errors in this collection. They forgot to shim, they switch between samples and they do not reshim and so on. And these users can use this program just as a black box to correct the spectra. That's the idea.
Okay, thanks. Another one, it's about the Magnetstein. What with the where we have, for example, moving peaks or changing J-couplings?
Well, J-couplings don't matter because there are no peaks in the method. There are just piles of sand. So
but it's different.
The peak can be split into two. It can merge again in the series and so on. Doesn't matter. So depends whether we talk about this application to the mixture analysis or to reaction monitoring. But in both cases, you may, for example, have a peak that is well-resolved doublet in the library, but in your mixture, this doublet appears as a singlet because of some inhomogeneity. It can even appear as a singlet that is slightly shifted. It doesn't matter. What matters is that, well, you have a pile of sand close to the pile of sand in the library component. And the advantage of the Wasserstein distance compared, for example, to the Euclidean distance is that it is sensitive to the peak shift. So if a given peak is shifted by, let's say, 0.1 ppm between library and the mixture, the Wasserstein distance will be different than when it is shifted by, let's say, 1 ppm, while Euclidean distance would be the same. So for simple difference between spectra, like L2 norm of the difference, it will be the same whether the peak is shifted by 0.1 ppm or 1 ppm. The conventional fitting of your spectra with the combination of spectra of the library components is insensitive to peak shifts. The answer is that it doesn't. The Wasserstein doesn't even think about J-coupling. So, of course, the more the peak is deformed or the more it is shifted, the Wasserstein distance is bigger, and well, the optimum of the peak is shallower, so to say. But it's not that it is somehow more sensitive to, or that changes in J coupling somehow prevent the application of Wasserstein in any specific way. So it doesn't matter. It doesn't even know that there are J couplings or peaks or anything.
Okay, thanks.
Any other questions? Doesn't look like that. Okay,
May I ask a question?
Yeah,
Yes.
of course.
About this ShimNet, is it going to be an open source like the Magnetstein is? Okay,
Yes,
so
yes.
it's on upcoming, yes?
Yes. It is upcoming. As I said, it requires some effort during calibration. So you need to have a machine with GPU. You need to, of course, have all these libraries for machine learning and so on. But once it is trained, it is very easy to use. And it's also super fast. So it is like in a fraction of the second, it corrects your spectrum.
So basically we will have to register the training data for one sample and then process them. Okay.
Yes.
Okay.
And it will be all described in GitHub. Actually already is, but it's not public yet.
Okay, thank you very much.
Thank you.
Anyone else? Okay, let's wait a moment. Okay, I think the questions are over. So I'll change the slides once again. Share. So I'd like to invite you all for the next seminar in 2 weeks, which will be given by Dr. Aneta Lennart from Technical University of Munich. And she will talk about deciphering the role of post-translational modification as key regulation of disordered protein regions using NMR-based structural biology approach. And then we'll have another seminar. So we have a queue till middle of May. I invite you still to send me some proposal for another seminars. And thank you all for your attention. Thank you, Krzysztof, for participating in this seminar as the first speaker. This is always the hardest. And see you all in 2 weeks.
See you. Bye. Thanks.
Thank you very much.
Thank you.
Bye-bye.
Thank you. Bye.
Thank you.